Today we are going to review what we've learned about mitosis, learn about what causes cancer, and then learn about meiosis. First of all, let's talk about cell growth. In most cases, living things grow by producing more cells. And there's two main reasons why cells divide. First of all, the larger a cell gets, the more demands it places on its DNA. It's only got so much DNA that it can use at a single time, and so it can only make so many things at a time. It's got to kind of wait in line and wait its turn to use the DNA. Then, as the cell gets larger, it also has more trouble moving enough nutrients, enough food, into the cell, and moving enough wastes out of the cell across the cell membrane. If you're interested, here's an explanation of why that happens. Before a cell gets too large, it divides, forming two daughter cells. Cell division is the process by which a cell divides into two new daughter cells. Now let's take a look at the cell cycle. As you already know, a cell must copy its genetic information before cell division begins. Each daughter cell then gets a complete copy of that information. So in most prokaryotes with no nucleus, bacteria, cell division, there's no nucleus, so you just cut the thing in half. But in eukaryotes, cell division includes mitosis and cytokinesis. Mitosis is what divides the nucleus. Cytokinesis divides the cytoplasm and everything else in the cell. And the cell cycle is how the cell goes through the steps of living and then dividing. During the cell cycle, a cell grows, prepares for division, and then divides to form those two daughter cells. And each daughter cell then begins the cycle again. The two phases of the cell cycle include interphase, which is when the cell's just growing and living and acting like a cell, and cell division, which is made up of mitosis and cytokinesis. Most of the cell cycle is spent in interphase, approximately 90% for most cells. And interphase is divided into three phases, G1, S, and G2. During the G1 phase, cells increase in size and make new proteins and new organelles. It's just living. Think G stands for growth. I think it actually stands for gap, but growth will make a whole lot more sense in your head. In the S phase, the chromosomes are replicated. They're copied. You make an exact copy is a replica. And S stands for synthesis. You are synthesizing a copy of the chromosomes. You are making a copy of the chromosomes. Then, during G2 phase, the cell continues to grow, plus it prepares for cell division. Next, the M phase, or cell division phase, includes both mitosis and cytokinesis. Mitosis makes two twin copies of a cell. Here are all the T's. Mitosis, two twin cells. That's how you can remember that. The two daughter cells are identical to the parent cell. Then, mitosis is divided up into four phases, which you've already learned. Remember, good old PMAT, P-M-A-T, prophase, metaphase, anaphase, telophase. First of all, during prophase, the chromosomes condense or coil up and become visible. And there are two centrioles which separate and move to opposite sides of the nucleus. And the centrioles make the spindle, which makes the spindle fibers, and that moves the chromosomes apart later on. During prophase, the chromosomes just attach to the fibers in the spindle. And at the end of prophase, the nuclear membrane breaks down. Number two, during metaphase, the chromosomes line up across the middle. Middle. The centromere of each chromosome attaches to the spindle fibers that came from the centrioles. During anaphase, the cell moves the chromosomes apart. Anaphase apart. The centromeres that join the sister chromatids split, and the sister chromatids become individual chromosomes, and the two sets of chromosomes move apart. Remember, it's a chromosome when it's a pair of sister chromatids, and it's still a chromosome when it's just one sister chromatid. It's a little bit odd. Finally, step 4, during telophase, the chromosomes gather at the two opposite ends of the cell. Tele splits in two. You will actually see that split forming in the middle during telophase. The chromosomes gather at the two opposite ends of the cell, and they lose their distinct shape by uncoiling back into chromatin rather than chromosomes. And at the same time as telophase occurs, cytokinesis also occurs. Remember, cytokinesis is cell cutting, and if there's a plant cell, a cell plate forms halfway between the two nuclei. One nucleus, two nuclei. And then a cell wall forms inside the cell plate. Then in an animal cell, a cleavage furrow, a ditch forms, and it pinches the cell in two. And remember, mitosis divides the nucleus and the chromosomes. Cytokinesis divides the rest of the cell, the cytoplasm and the organelles. All right, let's look at some new material. In a multicellular organism, cell growth and cell division are carefully controlled. For instance, when an injury such as a cut in the skin occurs, cells at the edge of the cut divide rapidly and keep multiplying until the two sides of the cut finally touch. When the cells touch in the middle, they stop dividing. They just go back to, okay, I'll replace a cell here and there, but not dividing rapidly anymore because they feel the crowding. It turns out that there are cyclins, that are a group of proteins, that regulate the cell cycle, and there are regulatory proteins that control the cell cycle. And these controls can be turned on and off, kind of like when you get a cut. Turns the controls off and lets the cells grow. Then it turns the control back on and it controls it and stops it, and the cells quit multiplying so fast. But what if these controls don't work? That's cancer. Cancer is a disorder in which some of the bodies lose the ability to control growth. Cancer cells don't respond to the signals that control the growth of most cells, and as a result, they divide uncontrollably. We'll learn more about cancer in a later assignment. Finally, let's look at meiosis. All right, mitosis makes two twin cells, two identical cells. So for example, if you cut yourself, then it's going to make more skin cells and seal them back up. You don't want it making different cells, stomach cells, brain cells, that would be bad. So you want it to make identical cells. But meiosis make eggs and sperm. I remember the letter O looks like an egg, and I guess if you added the tail, it would look like a sperm. So meiosis, and it is not meiosis, it's meiosis. That makes eggs and sperm, and each of those has half the usual number of chromosomes. It actually makes 4 daughter cells rather than 2, and each of these daughter cells are different from the parent cell and different from each other. This is how a baby ends up looking different from either one of the parents. You probably already know that living things inherit a copy of each gene from each parent. So you might get a copy of a gene for blue eyes from your mom, brown eyes from your dad, and then whichever one is dominant, that's the color of eyes you have. These copy of the genes are separated when the gametes form. So your mom will pass on just one of her 2 genes, and your dad will pass on just one of his 2. And then you may end up looking similar or different depending on which gene was passed on. Your parents have 2 copies of each gene, but they only pass on 1 each to you. So these copies are separated when gametes form. What's a gamete? It's a sex cell. What's a sex cell? Eggs and sperm. The process in which eggs and sperm are made is called meiosis. Meiosis is a process in which the number of chromosomes per cell is divided in half. That way you get half your chromosomes from your mom, half from your dad, and you still have the normal number when it's all done, instead of having twice as many, which would be bad. So this happens by the separation of homologous chromosomes in a diploid cell. Clear as mud, right? A pair of chromosomes that controls the same genes is a homologous chromosome. One homologous chromosome comes from your mom, and one comes from your dad. So you have 2 copies of each chromosome. Those are homologous chromosomes, the 2 copies. They control the same genes, but they're not necessarily identical. For example, you can have 2 genes for eye colors, 1 gene from your mom and 1 from your dad, but those genes may not be the same. You may have a gene from your mom for blue eyes and 1 from your dad for brown eyes. That's why they're homologous, not identical. Now, what about the diploid part? Having a pair of each chromosome makes it diploid. That's considered to be 2n total chromosomes, 2 times the base number of chromosomes. And having a single copy of each chromosome makes a cell haploid, and that's considered to be n total chromosomes. So for humans, we have 23 pairs of chromosomes. N is 23. But each of your cells has 2 copies of each chromosome, 2n. So 2 times 23, we have 46 total chromosomes. 23 pairs, 46 total. So when a person makes eggs or sperm, they start with a diploid cell, 46 chromosomes, 2 of each. But each egg or sperm only passes on 1 of each chromosome. So the egg and sperm are haploid. They have half the normal number of chromosomes. Then an egg with 23 chromosomes, a sperm with 23 chromosomes get together, fertilization, and the baby that results has 46 chromosomes, just like he or she is supposed to have. So a cell with both sets of homologous chromosomes is diploid. Di means 2, like you roll 2 dice. So a diploid cell has 2 sets of chromosomes, 1 from mom and 1 from dad. Somatic cells, body cells, are all diploid. A cell that has 1 set of homologous chromosomes is said to be haploid. Haploid have half the number of chromosomes. Only gametes are haploid. What are gametes? Sex cells or egg and sperm. Okay, so eggs and sperm are the only haploid cells. Every other cell in your body, every other cell is called a somatic cell, is diploid. Meiosis looks a lot like mitosis, except it's kind of like it happens twice. Since it divides twice, that's how you get 4 cells rather than just 2. And before meiosis begins, the cells undergo DNA replication. They form duplicate chromosomes. But wait a minute, if a cell's going to divide, then why would it want to copy its chromosomes and have twice as many and then divide twice? What's going on? Well, the reason it copies its chromosomes and makes twice as many first is so those 2 copies can trade DNA so that the baby ends up different from the parents. In a minute, we're going to talk about alleles, and I just want to mention that an allele is a version of a gene. For example, you can have an allele for brown eyes or an allele for blue eyes, but both alleles are for eye color. They are both versions of the gene that controls eye color. Okay, so then the cell goes into actual meiosis, and it actually does PMAT twice. In meiosis 1, it does prophase, metaphase, anaphase, telophase, and 2 cells form. However, each cell has sets of chromosomes and alleles, versions of the gene, that are different from each other and different from the parent. This is because the chromosomes undergo crossing over, in which the chromosomes cross over and trade DNA. Let me show you what I mean. There's a picture at the bottom. When homologous chromosomes, 2 copies, 1 from your mom, 1 from your dad, they duplicate, they cross over each other, break off, and trade DNA. See how that works? Then they split again, and that's how you end up with 4 totally different gametes, 4 different sperm or a different egg than you started with. It turns out sperm make 4, but for an egg you want it to be really good, so it keeps the DNA. The DNA is split equally, so you don't have any extra DNA because that would be bad. But then all of the cytoplasm and the organelles go into one of the eggs, and the other 3 pieces of DNA go into this little like throwaway egg that's called a polar body. And it never gets used, it's just tossed. That way you have 1 really good egg and 3 that you just toss and your body doesn't use. So we are down to splitting once. So now we have a group of DNA and a group of DNA. Then it does meiosis 2, PMAT again. The only thing is the DNA is not copied first. So now we split the DNA, now we have half as many chromosomes. So the cells divide again, PMAT, prophase, metaphase, anaphase, telophase. Now we have 4 daughter cells, and each daughter cell contains half the number of chromosomes as the original parent cells. And these 4 daughter cells are gametes, 4 sperm or 1 egg and 3 throwaways. Let's take a look at meiosis. So here are your chromosomes, and they double. They're replicated. But instead of making 1 single file line, they make 2 lines for meiosis. They split, PMAT, but they don't double again, then they just split again, PMAT again. That's meiosis 1 and meiosis 2. And then you end up with either 4 sperms, or, like we said, 1 very large egg and 3 polar bodies that just have the DNA and a tiny bit of cytoplasm, and your body just throws them away and doesn't use them. So mitosis and meiosis have a pretty similar sound. A lot of people say meiosis to tell the difference between mitosis, but that's incorrect. It's mitosis and meiosis. And so you just have to say them very carefully to tell the difference. Mitosis makes 2 twin identical cells. These cells are exactly like the parent cell. But meiosis makes 4 cells with half the number of chromosomes, haploid, and they're genetically different from one another, so the baby will turn out different.